So good afternoon, everyone. Welcome to our cardiology grand rounds. As you know, this is an hybrid event, so we welcome also the audience that joined the event on Zoom. And also we have people on site from our clinic. For the people on Zoom, at the end of the lecture, there will be a question and answer session. So please either speak up or raise your hand, and welcome from our side. So today we have Frank and myself the privilege to host Professor Mauro Giacca. It's really an honor for us. Thank you for being with us. I've seen your talk a couple of years ago in Lausanne. I was pretty inspired when I told Frank we should have Mauro in Zurich. So you don't really need an introduction. I would just say a couple of words about your outstanding work. Professor Giacca is the head of the School of Metabolic and Cardiovascular Medicine and Professor of Cardiovascular Science at King's College, London. He is the past president of the International Society of Heart Research. He's the elected fellow of the Academy of Science of the United Kingdom, and he is founder of 2 startups that develops cardiovascular biologics. Professor Giacca has an interest in cardiac repair and regeneration and was a pioneer in this field. He has done terrific work to develop new cardiovascular biologics to boost cardiac repair in the context of ischemic cardiomyopathy and in the context of heart failure. He was among the first to identify microRNA which have the ability to induce cardiomyocyte proliferation in the mammalian adult heart. And this was a major breakthrough from his work. He has done outstanding work and received prestigious funding from the ERC. He received 2 consecutive ERC Investigator grants, 2 consecutive LEDUC, and also a big grant from the BHF research program. So that having said, I leave you the stage, and thank you again for being with us, Mauro. We very much look forward to it from you. Thank you.
Okay, I was saying thank you very much for having me here, Francesco and Frank. It's really a privilege for me to be here. And thank you also for the very generous introduction. So this is my disclosure. They are mostly related to 3 companies that I funded since I moved to London. I start the presentation with this slide on the phase 3 pivotal trial that led to the clinical approval of sacubitril/valsartan for HFrEF and HFpEF, and for the glyphosines, again for HFrEF and HFpEF. You're certainly well familiar with these results. The reason why I'm showing this here, it is to draw your attention to the difference in the efficacy curve for treated and standard of care, which is so small, this difference, that it needs to be enlarged in the inset of the figure. And these trials were performed from a minimum of 5,000 to a maximum of 30,000 patients for 5 years observation to reach a statistically significant p-value. And this is drawn to the attention to the fact that really we are dealing with an incremental effect of all the therapies we have available for heart failure. A concept that a few years ago led to Milton Parker, who at that time was the president of the Heart Failure Association of Failure Society of America, to write this editorial on the impossible task of developing a new treatment for heart failure. And if you apply this concept, especially to HFrEF, the reason why I believe there is such a difficulty of finding new therapies that are effective in patients and decrease mortality. Let's consider that 20 years ago, mortality after diagnosis of heart failure was approximately 50% at 4 years, and it is 50% at 4 years still now, despite all our advances. The reason why there is such a difficulty is because really we're dealing with a condition in which there is a loss of cardiomyocytes, so in which basically the amount of cardiomyocytes we have in the heart of patients is decreased compared to normal. And there is evidence of cardiomyocyte loss in virtually all conditions of heart failure with reduced ejection fraction. So obviously there are these loss of cardiomyocyte after myocardial infarction. Upon approximately 1 billion of cardiomyocyte out of the 4 billion, the left ventricle can die in acute myocardial infarction. But basically all cardiac disease kills cardiomyocytes. There is cardiomyocyte death in hypertensive heart disease. There is cardiomyocyte death in all condition of inherited cardiac disease. Myocarditis and virus infection kills cardiomyocytes. Doxorubicin and other oncological treatments kill cardiomyocytes, and aging itself depletes cardiomyocytes. And the problem of the heart is that it is a post-mitotic organ. So basically, you can take out cardiomyocytes from an adult heart. You have these beautiful cells. They are very big simply because from birth to adulthood, the heart increases in size, but not because cardiomyocytes divide, but because the cytoplasm increases in size. So they become hypertrophic. 10% in humans are binucleated. You can do whatever you want with these cells in culture. You can do fantastic immunofluorescence, electrophysiology, molecular biology. You can transfer genes, but these cells won't divide. And there is converging evidence now from genetic studies in mice and from investigations in humans that the capacity of cardiac regeneration, so formation of new cardiomyocytes during the adult life, is less than 1% per year, which means that a person who is 70 has more than 50% of his or her heart, which is formed exactly by the same cells with which she or he was born. Just to give you a glimpse of this incapacity of the heart to regenerate after birth, this is a new genetic system that we developed recently in the laboratory, in which we can label specifically cardiomyocytes that replicate. And this is based on the Cre recombinase under the control of a promoter which is activated only in mitotic cells. This is a 312 base pair segment, which gets transcriptionally activated only when cells are in G2 or M. It is very, very specific for this period of the cell cycle. And we use this in the form of an AV vector to go specifically to cardiomyocytes in mice in which the GFP gene is floxed, so it is inactive, and becomes active only when Cre is active. So basically cells that become green are cells in which replication has occurred, and then these cells remain green for the rest of their life. And basically we compare the vector with this construct here, with a vector in which Cre is instead under the control of the cytomegalovirus promoter. So basically a promoter that is always active. And these are the results in neonatal mice. And so where you see green cells in this part here, it is green cells that have been transduced with an AV vector in which Cre is under control of the CMV vector, so CMV promoter. So basically all transduced cells become green. And here instead we use this replication sensitive promoter. And you see that in neonatal mice, there is a very large percentage of cells that are in active proliferation. More than 35–40% of cells are in the cell cycle immediately at the day of birth in mice. If you use exactly the same vector, the same doses in adults, the cytomegalovirus promoter continues to be active. However, the replication-sensitive promoter is completely inactive. You can see some proliferating cardiomyocytes here and there or there. So something occurs at birth by which our proliferative capacity is completely blocked in cardiomyocytes. And what is this something that happens is probably a combination of different factors. At birth, there is an oxidative burst, because there is oxygen directly coming from the lung to the heart before birth. The heart is a venous organ in the womb of the mother. At birth, the heart has to pump against a strong afterload, while before birth it is the mother heart that sustains most of the circulation. And these are probably contributing factors by which there is a signal that tells cardiomyocytes stop dividing, build up sarcomeres, and increase in the site. By the way, this is a very interesting topic for investigation, because if we understand exactly the molecular mechanism for this, we could intervene. But the bottom line is that if you look at the problem of heart failure under this perspective, one would think that we need therapies that protect against loss of cardiomyocytes. We need therapies that induce formation of new cardiomyocytes, but we don't have either of these. So we have nothing that can protect the heart and nothing that can regenerate the heart. And all the drugs that we have desperately try to make the residual cardiomyocyte work a bit better, either directly acting on cardiomyocyte or indirectly acting on the circulation. And obviously, it is very difficult to improve, and this is the reason why all the therapies that we have available have only this small incremental effect. There's been nothing transformational since the introduction of the ARBs in the 90s or the ACE2 inhibitors and beta blockers in the 70s. There's been nothing really transformational in the therapy of heart failure. And all the treatments that we have available are small molecules. It is super surprising that in a condition such as heart failure, which is the most prevalent condition in the world now, 65 million people worldwide in big metropolitan cities in Asia, in Africa, people die more of cardiometabolic disorder than all the viral or protozoal infections put together. It is very surprising that we only have small molecules. So basically, cardiology has not transitioned to developing recombinant proteins, monoclonal antibodies, and even less in the new area of nucleic acids, so RNAs, or viral vectors, so gene therapy, or cells, or three-dimensional tissue. And so this is the preamble where my laboratory starts operating. We want to find new biological drugs for cardiac protection and cardiac regeneration, and we want to find also new ways of delivering gene editing components for cardiomyopathies, but I won't speak about gene editing today. The problem is, since there's been so much failure so far, how can we find an effective drug? And if you think the way you can approach the problem of finding a drug, you have basically three alternatives. One is that you have an hypothesis. So you do hypothesis-driven research. And you say, I study a pathway, and within a pathway, I try to identify something that works, and I apply it to the heart. This is the way that has been done so far. A second approach is that you do omic studies. So you take pictures of your, for example, normal heart, your heart in heart failure, you see the genes that are differentially regulated, and you try to inhibit the genes or deliver the gene or overexpress and see and hope that someone will work. But nothing of this has brought to any therapy so far, any biological therapy. The third approach is that you forget about hypotheses, but you search for something that works. So you search for something that works first, and then you go back and try to understand how it works. So you do selection of screenings, which are the most systematic possible, and you do this in vivo. And in vivo for the heart, surprisingly, is relatively easy, because there is a family of vectors, AAV vectors, that are surprisingly good in post-mitotic cells. So exactly in cells in the body that don't divide, will never divide. So cardiomyocytes are one of these cell types, but other cells are retinal cells, both photoreceptor and retinal pigment epithelium cells, or neurons, skeletal muscle fibers. And surprisingly, AAV vectors work beautifully in these cells, while they work very badly in cells that replicate, for example, in tumor cells. Again, fantastic biology. We can speak about this later. If you take an AV vector that expresses GFP, you can inject it into the left ventricle directly, or you can inject it systemically. It will go both to the left ventricle and the right ventricle, and it will target only cardiomyocytes, not fibroblasts, not macrophages in the heart, not endothelial cells, only cardiomyocytes. So we have a tool to deliver genes specifically to cardiomyocytes. Then we thought, let's try to develop a method to select for cardioprotective factors directly in vivo in a functional way. So suppose you have a library of AV vectors, and each vector expresses a different factor. You inject this pool of vectors into the heart. Each vector will go to a different cardiomyocyte. Then you induce damage. You ligate the coronary artery, the LAD, and so a vast portion of the left ventricle will die. But if a single cardiomyocyte expresses a factor that is protective, it will be preferentially selected. So you do functional selection in vivo. We call this method FunCell. And functional selection means Darwinian selection. You select something because it is protected. And then you wait 3 weeks, you recover DNA from the heart, and each of the vectors has a barcode, so it has a 10 nucleotide sequence, which is unique for that factor. And then you do next generation sequencing of the barcode, you will have a certain number of reads for each barcode. So if a barcode is overexpressed, it means that the cardiomyocytes that express it are selected, and so that they are likely to be cardioprotective. If one is instead downregulated, it means it was deleterious. So we embarked in a project of constructing a library of the secretomes. So we scanned the genome and selected all genes that bioinformatically are known to be secreted outside the cells, either because they have a signal peptide or a conformation by which they are produced in the endoplasmic reticulum and then secreted outside. And so basically, we have a collection of 1,200 different factors that code for all cytokines, interleukins, growth factors, extracellular matrix proteins, everything that is secreted by the genome, not by the heart itself. And then we run this selection. This slide includes the data from over 800 mice and 1,200 factors in pools of 18 per mice. And we used pools of 50 vectors for the screening. And we found a series of factors that are super highly protective. Here, the axis is a z-score. So these are the standard deviations from the mean. So it means that there are factors that protected in this first round more than 5 standard deviations from the mean of all the others, and there are equally factors that are negative, that we took the top 200 and ran secondary screenings, and eventually ended up in a series of factors which are listed here that are really very protective. And we tested them one by one, and you see that they really protect the heart from death. These experiments were performed by permanent LAD ligation, so inducing a huge damage to the heart without even reconsidering reperfusion. And so it creates big infarcts in the controls. And despite the permanent ligation, you see that there is a very considerable portion of the heart that is protected, especially with these 3 factors. They're called cordon-like 1, FAM3C, and FAM3B, completely unknown to the cardiac literature. We would have never found them by studying transcriptomics or hypothesis-driven research, because they are not expressed in the heart. Well, FAM3C is indeed, but didn't come out in any of the hypothesis-driven research. They are completely new to the cardiac literature. Cordon-like 1 is a protein that is expressed during development. It is an inhibitor of BMP4, less affinity to the other BMP family members. FAM3C and FAM3B are 2 members of a family, the FAM3 family, which was described a few years ago because of their structural features. And FAM3B is also called PANDER, and it has some literature on beta cell function. FAM3C or ELE instead has metabolic activities in the liver. why did we select this factor specifically? Well, the first reason is that they are strongly anti-apoptotic. So basically, if we measure by tunnel apoptosis in the heart 2 days after myocardial infarction in mice, all these red dots are nuclei of apoptotic cells. But if the heart is immediately treated after myocardial infarction with cordon like 1, fan3C, and fan3B, the amount, the number of apoptotic cells is very massively reduced, especially for cordon like 1, fan3C. Really, you drop this to 0. And something that we didn't expect, and this is the reason why we selected them, because we are selecting for viability, and then these cells block death. These factors block death of the cells. Something we didn't expect is that all these factors are strongly pro-autophagic. So they stimulate autophagy. There is a strong induction of autophagic protein, LC3 translipidation, ATG5, and there is an induction of the whole flux of autophagy. So here, with this factor, remember that we inject our AAV vectors immediately after ligation of the LAD. With these factors, from AAV vectors, expression occurs after 4 hours and then peaks 12 hours in conditions of damage in the heart. So we are basically protecting cardiomyocyte in a window from the first 4 hours to the first 2-3 days, when some literature says that there are up to 70-80% of cardiomyocytes which are indecisive whether to die or not. So basically, we are helping survival of a large portion of cardiomyocytes that are damaged by the infarct, but still are alive for a few hours after myocardial infarction. And a lot of the pathology in this window comes from dysfunctional mitochondria. Mitochondria usually channel through the production of reactive oxygen species, 0.2% of the oxygen they burn. But after myocardial infarction, this percentage can rise to 2%, 3%, 4%, a lot of damage in mitochondria. So if you remove mitochondria, basically you save the heart. We know also some additional activity of cordon like 1. It is a strong BMP4 inhibitors, and we know that after myocardial infarction, there is a BMP release, BMP4 release, which is detrimental, and it impairs autophagy. And BMP4 inhibitor, it also inhibits a cognate family member, which is TGF-beta. And so basically cordon like 1 has a dual role in BMP4 and TGF-beta. And in fact, all the hearts, I remember that when we did this experiment, the postdoc that were doing this experiment always came to my office saying the hearts of the animals in which we did myocardial infarction and treated with cordon like 1 remain much smaller. And we wanted to look at this, and there is, in effect, much less fibrosis. This is a transgenic mouse in which green is collagen. You see this is the control, a lot of fibrosis in after myocardial infarction. You see much less fibrosis if these hearts express cordon like 1, and also in vitro, we can trigger myofibroblast formation by TGF-beta. This is SMA staining, smooth muscle actin staining of cardiomyocytes, where it's red, it is myofibroblast formation. If you give cordon like 1, in this case it is recombinant, 3 doses, you see that you massively decrease this. So we selected in our method factors that have this combined activity. I believe we will never found them simply by studying the literature. How can we transform this into therapeutics? Well, first we did an experiment in which we wanted to prove that they work also as circulating factors. The reason why we did a library of the secretome instead of a transcription factor or other categories of intracellular proteins, it is because we don't want to stick to AAV vectors for their delivery. We don't want to do gene therapy. We want to give a factor that can be injected intravenously and reach the heart through the circulation. And obviously, a protein that works extracellularly has these properties. So we did a proof of principle experiment in which we built again an AV vector, but this time with a liver-specific promoter, and package it into an AV8 vector that goes well to cardiomyocytes and not well to liver cells, not as well to cardiomyocytes. And we injected it into the liver. There is nothing of the vector in the heart, but the protein is secreted in the liver. This is so good to be secreted that we can see it circulating in serum. And this is very protective too. Big infarcts in the absence of treatment. And this is circulating cordon-like 1, FAM3C, and FAM3B, much reduced infarcts with all the correlates of cardiac protection. And on the basis of this these findings, we were successful in convincing a large venture capital company in London in investing in the creation of a startup company that's called Forcefield, which is generating recombinant protein. These are some of the first results in which basically we infarct the mice and inject the recombinant protein for FAM3C and FAM3B immediately after myocardial infarction, and then look at what happens after 1 month. This is ejection fraction in the treated animals compared to control, and you see that there is improved ejection fraction, reduction of dilatation, so all the echocardiographic correlates of protection, and histology confirms this data. This protein now is being tested in pigs, preliminary to go into clinical experimentation in humans. There is probably another way of administering this protein, and this has been taught by the very recent success of the COVID vaccines. You know that the Moderna and the Pfizer COVID vaccines use a technology for the delivery of the messenger RNA for the spike protein based on lipid nanoparticles. Lipid nanoparticles are 100 nanometers or less in diameter spheres that are formed of 4 lipids. 2 lipids are helper lipids. Usually one is cholesterol, another one is phosphatidylcholine lipid. One is a lipid which is pegylated, and this gives stability. But the most important lipid is the 4th, which is a ionizable lipid. So a lipid that a pH 4 is positively charged, so it binds to negatively charged nucleic acids tightly. Then you raise pH, it becomes neutral. So you form a sphere which is neutral. You can inject it in vivo, and it's not inflammatory, doesn't activate the platelets or the coagulation system. And this technology is called the SNALP technology, stable nucleic acid lipid nanoparticle technology. Was first approved for clinical use with patisiran in 2018, which is a lipid nanoparticle with an siRNA against transthyretin for transthyretin amyloidosis. So it targeted transthyretin gene. It's one of the genes, one of the treatments that are now tested also for cardiac amyloidosis. And then it had all the success in the COVID, Pfizer and Moderna vaccines. So basically what we did was to try and develop lipid nanoparticles that target very well the heart, are not inflammatory. So basically here, this is the first set of experiments in which we used the EGFP messenger RNA, and we had normal mice. We injected intravenously with these lipid nanoparticles, and then after 1 day, we looked for cardiac, for fluorescence in the liver, and we tested the 3 different formulations to adapt them well to specifically expressing in the liver. You see that both 3 formulations are quite good. If you inject these particles systemically, they all go to liver and spleen, all the 3 of them. A bit in the lung. They are uptaken usually by the apolipoprotein E receptor, and which is also expressed a bit in lung cells. And there is almost nothing that goes to the heart or skeletal muscle. And we picked up the formulation that is less activatory of the innate immune system, so that recognizes these lipids. And then we did this by testing the expression of 3 genes, which are normally activated as part of the innate immunity. You see that this formulation here is the least immunogenic. And then we took the mRNA from one of these for our factors, and included this in lipid nanoparticle and injected it immediately after myocardial infarction. And we see that the protein is released in serum as early as 4 hours in some of the animals, certainly after 6 hours in all the animals, and then it peaks around 24 hours. It stayed there up to 72 hours. And a single injection is sufficient to protect the heart. You see that the difference in the size of infarct, these are 2 representative animals for section from base to apex of control animals and treated animals. You see there is much more muscle preserved here and all the correlates of cardiac function. So we believe that an alternative for cardioprotection is to have in a patient with myocardial infarction injected either the recombinant protein or a lipid nanoparticle with a messenger RNA that goes to the liver and secretes acutely a protective factor. This issue of cardioprotection is also very interesting in other settings, and one of these is obviously doxorubicin-induced cardiotoxicity. We tested first AV vectors for protection of cardiomyocytes against doxorubicin toxicity. You see that this is caspase 3.7 as a marker of apoptosis. There is increase of apoptosis, which is dose dependent with doxorubicin. If the cell express according like one, FAM3C and FAM3B, they are protected. What was striking is an in vivo experiment that we did with AV vectors, in which basically we treated chronically with the doses of doxorubicin mice, and then followed them up to 80 days, and 75% of the controls died. And in the animals treated instead with the coordinate FAM3C, 100% of the animals survived. So the difference is 25% survival, 100% survival in the animals that express in the heart these proteins. And obviously this is very interesting for cardio-oncology applications. And as I said, now Forcefield is developing these concepts to achieve a new generation of drugs that could protect the heart and save cardiomyocyte against loss due to either myocardial infarction or in cardio-oncology settings. Obviously, we don't want to limit these therapeutics to cardioprotection. We really would like to regenerate the heart. When cardioprotection fails, then let's try to make more cardiomyocytes. And here the field has been quite problematic. You know, the whole concept of cardiac regeneration started 20 years ago with the assumption that there might be some cells that act as stem cells, either from the bone marrow, or the stromal compartment, or for the heart itself. Now you know that there are no stem cells that can regenerate the heart. All these, what was published was half failure and half fraud, unfortunately, that sets back the field for 15 years. There are cardiac progenitors cells, but these are only in the first phases of cardiac development. Then these cells mature to cardiomyocytes. All the replication that I've shown you in neonates is sustained by cardiomyocytes themselves. So there are no stem cells there. There's been a program by AstraZeneca of developing cardiac progenitor cells as a source for cardiac regeneration, and it was dropped 2 weeks ago for the difficulty of obtaining these cells. So we can safely conclude that really there are no stem cell therapies that can be injected in the heart for regeneration. Obviously, there are stem cells in embryonic stem cells or iPS cells from which one can derive cardiomyocytes. This has been pioneered by Chuck Murray and by others. And you can obtain 1 billion embryonic stem cells or iPS cells and convert them into 1 billion cardiomyocytes, inject this cardiomyocytes, but these are highly arrhythmogenic. It is possible that this can be circumvented by forming spheres. There is a clinical trial performed by Fukuda in Japan with cardiospheres obtained from these cardiomyocytes, or by engineering the cells by knocking out ion channel genes. This is what Chuck Murray does, but it is quite complicated. And you can use this cell to engineer heart tissue. And there are more than 10 patients implanted by Wolfgang Zimmermann in Göttingen with this engineered heart tissue. But you understand that this is all quite difficult to translate for a condition such as heart failure that affects 65 million people in the world, 80% of which are in developing countries. And in addition, regeneration in the neonate or regeneration in animals that regenerate like salamander or fish doesn't occur through stem cell-derived cardiomyocytes or myocardial patches, but occurs through cardiomyocyte proliferation. So we want to find a treatment that stimulates proliferation of cardiomyocytes. And we started this again by hypothesis-driven research. So we took all the genes that are known to be in pathway that are activated during development, the Notch pathway, the beta-catenin pathway, or the Yamanaka factor, DAV vectors put after myocardial infarction to see if we could achieve cardiomyocyte proliferation regeneration. All failed. Nothing of this works. So we say, okay, we need a screening. We have to do this serendipitously. And so we have to do this without a priori assumption. And we have to search for regulators that are very pleiotropic. Think of these big cells with all the sarcomere assembled. They have been there, not replicating for 50 years. And now we want to push them in the cell cycle. We need to disassemble the sarcomere, re-express all the cell cycle regulator genes, push the cells to proliferate in physiological conditions and so on. And we realize that microRNAs are very good therapeutic molecules because they have a pleiotropic effect. So each microRNA can target tens or hundreds of different genes, and then you can test them in vitro by high throughput screenings. And we started this project quite a few years ago, and to make the story short, we screened a library of microRNAs. For microRNAs that can put cells into the cell cycle, we found 4 microRNAs that are particularly active at proliferation, and then we use the genes of these microRNAs inside AAV vectors to see if we can regenerate the heart. And this is an experiment we did at that time. They are very, very effective. So basically, these are infarcts in the control at 12 days and 60 days, big infarcts, again, permanent ligation of the LAD. If you give the AV vector the microRNA, these infarcts are very significantly reduced. I think this was the first demonstration that you can really regenerate the heart. More recently, we applied the system for tracking or replicating cells to this to look at really the regenerated area. And these are the recent results that we have obtained. So this is a heart with myocardial infarction in which we injected the control AV. So all this is the infarcted area. And this is a heart of cognate animals in which we injected a vector for 199A or 590, the 2 top microRNA at that time. And this is the regenerated portion of the heart. And you can see that really you regenerate the heart through proliferation of cardiomyocytes. Consider that in this experiment, the efficiency of transduction is 40%, 50%. So you can even double the number of fluorescent cells at least 2 times. It gives you an impression that you really reform the muscle. We have now extended these screenings by more systematically. So we screened a library of 586 siRNAs that code for E3 ligases of the ubiquitin proteasome system, or 400 siRNAs that code each of which against a gene in the cytoskeleton sarcomere or 1,700 siRNAs, also including metabolic genes. So we have a series of at least 10 different small RNAs, each of which either microRNAs or by targeting a specific protein can push proliferation. And they're very, very active. Here, proliferation is seen by incorporation of BRDU. So there are these red dots here. This is the control. These are neonatal mouse cardiomyocytes. And these are the same cardiomyocytes treated with some of these siRNAs or microRNAs. See how many replicating cardiomyocytes you have. For 199A, we also went to pigs and did a similar experiment as we did in mice by injecting, this time is this ischemia reperfusion, injecting the AAV vector expressing 199 in 20 injections in the infant border zone. And then we follow these mice over time, these pigs over time. And these are the results at 1 month, and they are really striking, beyond all our best expectations. So this is infant size in black. It is the control animals. So sorry, in white are the control animals. You see that infant size remains stable from day 2 to day 28. And why it's significantly, massively decreased in the animals that express the microRNA. Ejection fraction it goes down in white in the controls, while it is up to levels that are not even statistically significant, yet different from non-infarcted animals, with all the correlates of reduction of fibrosis, or what's called remuscularization. You can see these MRIs, this is a control at 1 month, so the dilated left ventricle, thinning of the septum, and this is the remuscularized heart. So it works beautifully. And you see really massive cardiomyocyte proliferation. This is staining with Ki-67. These are the controls, and these are the treated animals. You see these big cells with the Ki-67 positive nucleus, and which is something that is really stunning. And this is staining for mitosis, so it's an antibody against phosphorylated histone E3. You see a lot of proliferating cardiomyocytes in the infra border area. And if it were like up to this is 1 month, we would have been happy, published our paper and stopped there. But we made a mistake. We left the animal alive to see what happens. And the animals, when they reached around 2 months, they started to develop arrhythmias. Several of them died. And in a few of them, we found the reason for this death and their arrhythmias, which was the hyperproliferation of cells that are never found in the heart. So there are cells that are very proliferating, and they express markers, high levels like GATA4, which is a cardiac transcription factor, but it is expressed at high levels during embryonic development, and then it drops down. Here, these cells are hugely positive. Or myogenin, which is a marker that is not expressed in cardiac cells, it's expressed in rhabdomyosarcomas, so aggressive tumors of the skeletal muscle. So bottom line, from AV vectors, you cannot control expression over time. So you have continuous expression of these pro-proliferative factors, which is good up to 1 month, but then obviously becomes deleterious. Here, however, we are lucky because we can circumvent the problem, because we are dealing not with the messenger RNA that we would need to have it constantly, but with the small RNA, which we can obtain as a synthetic molecules and give for just a short period of time. The field of small RNA therapeutics has boomed over the last few years. In this category, we include antisense oligonucleotides against messenger RNA, link RNAs, or microRNAs, and RNA interference therapeutics, so siRNAs and microRNA mimics. And I prepared this table for a review 2 years ago with ISHR, and at that time there were 9 antisense and 4 siRNAs that were approved for clinical use. And in July last year, this became 10 and 6, and now there are 12 and 7, just to give you an idea of the rapid pace at which these molecules reach approval. There are hundreds of small non-coding RNAs in different phases of the clinical testing pipeline. The problem is that if you use antisense, you can inject them systemically, and they enter the cells without having to use any delivery method. So just as naked modified RNAs, they can enter the cells. Oh, unfortunately, only less than 5% reaches the heart, but for some applications, this could be useful. However, if you use double-stranded RNA, like siRNAs or microRNAs, they don't enter the cells as naked molecules. You must facilitate their delivery. And obviously here again, lipid nanoparticles come very, very handy because you can use them to deliver this form of RNAs. And so we spend a lot of time at finding the right formulation. This is a microfluidizer where we do our preparations. The right formulation that works well in cardiovascular. Eventually, we found one that is very, very effective. Look at how many proliferating nuclei we have. This is always with 199. And this lipid nanoparticle can transfect more than 90% of cardiomyocytes in culture. This is the same formulation with the GFP mRNA. So all these green cells are cardiomyocytes that are transfected with these lipid nanoparticles. We were super excited. A PhD student spent 8 months at finding the right formulation to transfect cardiomyocyte, and was very excited, injected it in vivo, and it was a complete failure. So what we discovered is that you cannot develop lipid nanoparticle for in vitro application or in cell applications because the way they behave in vitro is completely different from the way they behave in vivo. So this is the same particle they gave almost 100%. It gives one cardiomyocyte here or one cardiomyocyte here. This is an antibody against the GFP. And so we started a tour de force to test directly and find lipid nanoparticles that work in the cardiac muscle. This is another ionizable lipid, a bit better, but still very unsatisfactory. We learned a lot along this journey. So we have now lipid, wow, here I don't see anything. Perhaps a bit more here. This is a lipid nanoparticle with GFP. GFP is seen with an antibody in red, which transfect very well the myocardium, but you notice immediately that none of these is a cardiomyocyte. So this is a lipid nanoparticle that you inject in the myocardium and it all goes to non-myocytic cells, but they are probably fibroblasts. Or this is a lipid nanoparticle that you inject in the heart and only goes to vascular cells. Here you don't see fibroblasts, you don't see cardiomyocytes. So there is something, it is a biology that has completely to be understood. So nobody knows what is the pathway of internalization of these particles, but eventually we found lipid nanoparticles that are quite effective in cardiomyocytes like these ones. And in particular, this one here, this is a single injection in the left ventricle. I'm sorry that the light is a bit high, so I don't know if you can see, but single injection, you see that it distributes quite well. This is a magnification, perhaps a bit better, in the cardiac muscle. And these particles also work in pigs. This is an experiment that we do in Pisa with Fabio Recchi and his group. And here GFP is seen with an antibody in red. And you see that we can transfect well also the pig myocardium. And in the meantime, we engineered with chemical modifications our payload, so the microRNA, by which we can introduce on the RNA backbone, which is here in gray. We introduce modification, especially in the passenger strand, which is not used for RNA interference, and we substitute several nucleotides with this chemical modification, and these chemical modification permit the microRNA to be present for more than 12 days, 12, 15 days, which is a sufficient period of time by which we have the replicative regenerative effect, but we don't enter into this hyperproliferation that we saw with AAV vectors. This is one of our nanoparticles. This nanoparticle also included some basic polymers. They have a size which is around 100 nanometers. We inject them in the heart and they stay there for 15 days. And sorry, I don't have the results here. Well, a single injection is sufficient to have reparative effect in mice, and we are now testing this also in pigs. We are obviously would have liked a lipid nanoparticle that can be injected systemically and reach the heart. Unfortunately, there is no way of doing specific cardiac targeting so far, but for the heart, we believe that we can achieve targeting by catheterization, because the heart can be reached through the coronary artery and through the coronary sinus, or intramyocardial through a mini thoracotomy, or less invasively through transendocardial delivery after left ventricle catheterization. So now a second startup is trying to develop a lipid nanoparticle with the formulation that we have discovered with one of our microRNAs to be injected at the end of PCI, with the idea of driving repair after PCI through remuscularization, so proliferation of cardiomyocytes, rather than leaving fibroblasts to proliferate. We had one problem still not understood, and the results are not complete, but I'm going to end by showing these to you to see if you share my excitement. It is, okay, proliferation, but you are working with mice which are 2 months old, which is adult, but still it's 2 months old, or pigs which are 8 or 1 year old, so they're still juvenile. Will a cardiomyocyte from a patient who has been for 50 years, 60 years, 70 years not replicating, be able to re-enter the cell cycle? And in addition, in condition in which this patient has heart failure. And obviously the question is very difficult to answer, but we tried at least to give a preliminary answer by developing in the laboratory a technology that permits to produce primary myocardial slices from human samples. And these live myocardial slices are 300 micron thick slices that are generated from different specimens, which could be explanted hearts, so patients that undergo cardiac transplantation of myectomies. And so the two persons who originally developed these techniques in Cesare Terracciano laboratory at Imperial now work in my laboratory, Filippo Perbellini and Sam Watson. So they established this in my laboratory, and we discovered that these slices can be easily transduced with AV6 vectors. Here GFP is in violet. My people like to change the color of how they visualize GFP. And these slices can be also transfected with lipid nanoparticles. And this is a lipid nanoparticle formulation with GFP, this time in red, in which we transfected these slices. You see that these particles are specific for cardiomyocytes, images that I find really very beautiful. This is a comparison of the efficiency of lipid nanoparticles with a commercial cationic lipid lipofectamine, the one that we discussed this morning. You see, they are really very, very efficient. They only go to cardiomyocytes. The exciting results, for which I don't have yet quantification, is this one. And this is a human myocardial slice from a patient who is 50-something, and who has heart failure to the extent that he was transplanted for heart failure a few weeks ago in the Heartfield Hospital in London. And we put his heart in culture in the form of slices and treated with one of our proliferative microRNAs, and you see that cardiomyocytes are able to enter the cell cycle. This is BRDU incorporation. And I think that this is the first demonstration I'm aware of that cardiomyocyte from a human patient in a diseased heart can still be pushed towards entering the cell cycle, which is, I think it is quite remarkable and sustains all our efforts in this area. So this was my last slide. Take-home message is functional screenings are very powerful to find new therapeutics. We developed a method called FunCell that permitted the selection of 3 novel cardioprotective factors, which can be used either as recombinant proteins or in the form of mRNA therapeutics. We can achieve cardiac regeneration by pushing proliferation of cardiomyocytes using microRNAs, and we can deliver all these RNA therapeutics using lipid nanoparticles. I love Banksy, and this is one of my favorite drawings, and I recently discovered that this drawing is on the Waterloo Bridge in South Bank in London, so not very far from where my laboratory is. But what's interesting is that this is in the context of this sentence here, says that there is always hope. And so I think that this can prove Milton Parker wrong, that it is possible to develop new therapeutics for heart failure. So a lot of people involved, you see them then in this movie. So all this work is performed in my laboratory in London. The screenings are by Miguel Mano. The AV vector is still produced in my previous laboratory in Trieste. Cardiac protection is heavily contributed by Francesca Bortolotti, lipid nanoparticles by Manendra Prachauri, together with Gang Lee. The myocardial slices are from Filippo Perbellini and Sam Watson. Cycle track, the method to see cardiomyocyte by Ilaria Secco, and I'm sure that I forgot all the other contributions, for which I apologize. And all the big work is performed in collaboration with Fabio Recchia. And I thank you very much for the invitation and your listening to this presentation.
Thank you very much, Mauro, for this outstanding, how would you define that, presentation and journey? I think also for our young colleagues, students, postdocs, it was very inspiring. So learn how to do stainings. So your lecture is open for discussion. We also have people online and on site. Please just speak up. Maybe, I don't know, Frank, if you want to initiate with a comment or...
Know where to start in such a extraordinarily fascinating presentation. You know, you're a young guy, so you're the future of cardiology. We've been talking about regeneration for so long. Now it's all about proliferation. Here's one thing. Thomas Thum was with us a couple of weeks ago on Grand Rounds. And not that he now apparently has sold his company, but that's not the thing. At that time,
1 billion, more than 1 billion.
Oh, yeah, something like that. And you remember Thomas was a couple of weeks and we had this discussed, and I was asking the same question and said, so he had just about to make, as you know better than I do, the way obviously into a phase 1 study or phase 2.
Phase 2,
yeah. And so how close are you to now applying this in patients? Because the obvious questions, you will always get them, is, well, stability, efficacy, safety. When you trigger proliferation, is that, even if it's specific in the heart, it's a good thing, but even there I see challenges.
Yeah. No, absolutely.
Arrhythmogenic substrate and all of that. With this, the specificity for the heart is one. Then even in the heart, do we want to have proliferation? That's and here's the thing for us, we are clinicians. How many cardiomyocytes do we have? Tchaikovsky, what do you think? I always ask him, he's very good. They're all good. They're all excellent. But however, have a guess? What do you think? Have an idea? 5 billion. Sorry? Oh, that's very good, actually.
Yeah.
It's 4
billion in the left ventricle.
Yeah. So it's, something like this. And within, just think about that. Next time when we have a STEMI coming today, you lose easily 1 billion cardiomyocytes. 1 billion. And they don't regenerate. So now just clinically, I'm sorry when I say that, but you want to think, okay, we can protect you. PCI is a little bit of protection. You just looked at the drugs, but they can't regenerate. So that's a pity. So that would be the ideal thing. And we will always come in some patients later. But do we want that to proliferate? Yes, I think yes. But that would not be
Yeah.
Without risk, because this is arrhythmogenic potential. And listen, safety is what is the key Achilles heel in this. I'm sorry when I'm...
No. This is very interesting. I think that the arrhythmogenicity is a misconcept because in the field, because if you think at regenerating conditions, so the fish, the amphibians, but also the human fetal heart, or the human, or say the animal fetal heart, all animals' fetal heart, and the experimental hearts at birth, as I showed you, there are 30–40% of cells that replicate, and there is not arrhythmia. And we discovered why. Using this cycle track, what you see that physiological replication of cardiomyocytes, suppose that cardiomyocytes are like that, no? They are this brick-shaped. So replication could be that these cells divide and becomes two cells, one on top and one on the bottom. This would mean that cells in the bottom should somehow intercalate into an array, an electrical array. This never happens. So replication is always of a cell within an electrical array. And this is even when we give our microRNA. And this is the reason why, for example, in pigs, we didn't see any arrhythmia for 1 month until this cluster of cells are generated, because replication is always in an electrical array, which means that if you trigger proliferation of endogenous cardiomyocytes, you don't have arrhythmia. But if you inject cells, you have arrhythmia, which is also proven by the cardiomyocytes that are injected exogenously.
Well, I agree with you, absolutely. This is not my concern. Actually, this is our concept as well, that when you look at this, the rate of sudden cardiac deaths did not go down with antiarrhythmic drugs. Mm-hmm. It went down when we started unloading the ventricle, and it got better. So that's the same here. If you will increase the efficiency of the heart, and through whatever it is, and the heart is performing better, you will have less arrhythmia.
Super good point.
The electrophysiology, I'm a heart failure guy who loves EP, but it's not that arrhythmias come out of the blue. They have a substrate or have a struggling ventricle. When you help it, that will be the best answer. I know that a lot of EPs will ask you, though, about arrhythmogenicity, which we have seen in drug development and then with small molecules all along. Yeah. But here it's different. I'm on your team.
Yeah. In terms of timing, for both applications, so both cardiac protection and regeneration, we are at the peak level. And the plan is to go to a phase 1 clinical trial in 2026. So we still have half of this year and next year to finish these experiments and do all the regulatory. The money is already there. So let's see. So it shouldn't be decades. Another thing about safety, the problem is would the microRNA do something else in other organs? And we know that they are safe in mice. We will know better if they are safe in pigs, and the microRNA that we have selected were selected to be anti-oncogenic in several different cancers. So, for example, this 199 is anti-proliferative in liver cancer, which is always a concern, in thyroid cancer, and in other cancer cell types. Then you will have to wait 15 days treatment once in a lifetime with the risk of having an adverse event, and then it will become another matter. So,
yeah, thank you very much, Mauro. Great talk. I'm a great fan. I was wondering whether you had looked at the particular niche where cardiomyocytes, if they regenerate or proliferate, are controlled in. So, I mean, that's a basic concept of the balance between proliferation and differentiation. The terminally differentiated cell, as a cardiomyocyte, should to some extent dedifferentiate, reacquire stemness, and these steps are controlled usually in defined niches. So it's a multi-step control process where niche cells, so fibroblasts and endothelial cells in many tissues such as tumors or immune organs, are taking part in the decision whether and to what extent a cell that used to be thermally differentiated, de-differentiates, and then reacquires the ability to proliferate.
The short answer is that we don't know if a subset of cardiomyocytes responds and another subset does not. What we can say is that if you, for example, provide our microRNAs to the heart and we use cycle track to map where it is, the area where proliferation is stimulated, for example, in neonates, is not different from the area where spontaneous proliferation occurs. It's a bit more towards the epicardium and the endocardium, a bit less within in the middle of the ventricles. I don't believe that the niche concept applies here because we know that the extent of de-differentiation is really very minimal. So just disassembly of the sarcomere and proliferation. So they don't go back to the embryonic state. There is in transcriptomics of cardiomyocyte after administering our microRNAs where we see proliferation, we never see any of the embryonic genes coming up. So they remain pure cardiomyocytes that, however, overexpress, re-express cell cycle genes, for example, and they downregulate a bit the levels of sarcomeric genes, but they remain cardiomyocytes that proliferate.
Thank you very much.
Yeah, and also proliferating, if you do, for example, this is a nice work by Eric Olson on neonatal regenerating heart, and by single cell sequencing, you see that there is a population of cardiomyocytes that proliferate, which is transcriptionally different from the other population of cardiomyocytes. But these are cardiomyocytes. These are they are cardiomyocytes that are very close to the other cardiomyocytes. They are not cells that become stem cells.
There is a question.
Yeah, there's also an online
Okay.
person. Caroline Leersmuller.
Okay. Yeah.
Hi, yeah, thank you so much for this.
Hi, Caroline.
Hi, thank you so much for this great talk. I have one very specific question because we often get asked about the disassembly of the sarcomere structure, which is something we very rarely see because we have been investigating the other, so endogenous exercise-induced cardiomyogenesis. And I think we can say that there is a limited number of cells that even endogenously proliferate and even divide, but very rarely can show the disassembly of the sarcomere. And I think you also showed it on only one of your slides. How do you try to see that?
I'm not sure I understood the question.
I think we lost part of the question.
So, Caroline, can you rephrase the question, please? Because we had a bad audio.
Sorry. So I was wondering how you try to show the sarcomere disassembly. This is something we often get asked, but
No, that's it. Well, when we published one of our early papers on cardiomyocyte proliferation, we showed the cardiomyocyte in vitro. Now we show a cardiomyocyte with all the striations, with an antibody against alpha-actinin. And then close to this, there was a cardiomyocyte that was expressing one microRNA in which the same antibody was stained diffusely the cytoplasm. And one of the reviewers said, so you have so bad stainings, look at it, it's so diffuse. And we reply, no, it is not diffuse, because what happens is that you have a disassembly of the sarcomere, but the sarcomeric protein stayed there. So basically, you can easily see that if you stain with alpha-actinin, troponin, and other components of the sarcomere, you don't see anymore the striations, but you see the protein diffuse in the cytoplasm. So my interpretation is that concomitant with cardiomyocyte proliferation, there is a signal to disassemble the sarcomere. And I hope that there is a single protein in the sarcomere that is responsible for this disassembly. The proteins are still there. What we are doing now is to block the proliferation stimulus and prove that the sarcomere is then reassembled. Because this is another problem, because you stimulate proliferation, but then you want also that the cardiomyocyte go back to their normal state.
Yeah, but this is in vitro, you said, right? But how often do you actually get to see it in vivo?
No, you see this picture also in vivo. let me, I just showed you this before, I didn't point them out.
Yes, I mean, we see it occasionally, but it feels like
Look at what happens. Can you see? Look at what happens, for example, in vivo. This is in pigs. You can look at the upper part. So you see here there are 4 nuclei that have divided or are dividing. So this is staining for phosphoinositide 3, which marks G2M. And you see that in the area where there is division, basically there is absence of the sarcomeric structure. So there is localized disassembly of the sarcomere. I think I have another one here. This is also very evident. So you have the cortical parts in which there is a sarcomere, but where there is division. This is probably not, certainly it's not cell division. This is nuclear division. So there is a multinucleation, which is very abundant. We have several of these pictures in pigs in which there is local disassembly of the sarcomere.
Yeah, thank you. Yes, it's just something I always get asked as well, and I feel it's so difficult to show.
Yeah, and by the way, these kind of pictures I discovered that were shown also in neonatal mice. If you look at the paper by Porello et al., the SADC Olson paper, the first that in Science, I think, described the proliferation of the neonatal heart, you will see pictures that are very similar to this on localized sarcomere disassembly.
Hi, thank you. That was a wonderful talk. I was wondering about the microRNA treatment with the proliferating cardiomyocytes. Are you also seeing a change in the secretome of these cardiomyocytes and how that might be playing a role in the fibroblast or endothelial cell populations?
No, good point. We never checked this systematically. The only experiment that we did quite seriously, it was to take the supernatant of the cardiomyocyte treated with microRNA, concentrate and put on cardiomyocyte to see if there was something secreted that mediates proliferation, and we didn't. So it seems to be cell intrinsic.
Thank you.
I have a question, please. Hello.
Or speak up and I repeat the question.
Oh, okay. So
Take this one. Come on.
Hello, now it works. Thank you very much for the great scientific contribution to the field. I really cherish your work. And so my question would be the big picture. So we have so many data also on cardiac protection for the last 25 years. We have so many concepts on cardiac regeneration. But despite all the good news, we couldn't translate it to the clinics. And what do you think, what is your bet on how we could accomplish cardiac regeneration and in what timeline? I personally think it's very complex and biology is complex and there are so many different compartments that contribute to cardiac regeneration. And do you think that just tackling cardiomyocyte proliferation will do the job to regenerate the heart?
No, very good comments. I would divide my answer for cardioprotection and cardiac regeneration. For cardioprotection, you are right. There have been many attempts and clinical trials and preconditioning, post-conditioning. I think that the focus has probably been not the right one because most of these experiments, especially the early ones, were performed in isolated hearts to look at the area risk, a few hours after looking at acute protection through mitochondrial function. And this has failed. What we are looking here is protection that occurs in the first 1, 2, 3, 4 days when there is a vast portion of. I think that those cardiomyocytes that die in the first 30 minutes because of acute mitochondrial dysfunction or whatever, they're dead, and we cannot protect, certainly not in injecting a vector. And why we protect all the other cardiomyocytes that are indecisive whether to die or not in the next hour, which are a considerable portion. And for cardiac regeneration, basically, it is true that the concept of cardiac regeneration started with Orlik et al. Nature 2001, so 20 whatever years ago. But it has been brought for 15 years based on false or fraudulent data. So that paper is retracted, and several other papers. We all know the story in the field. And while the idea of stimulating endogenous cardiomyocyte proliferation, I mean, it's relatively newer and takes time to go to the clinic, so there are no failed attempts. There is time to develop these attempts. For both of them, I would say that I think that one of the strengths of our approach is that for both applications, we don't start from hypothesis-driven research. I'm having a terrible, difficult life in the UK because UK reviewers and UK, the academic system, love hypothesis-driven research. So PhD students are taught to formulate an hypothesis, and then you do an experiment to prove that the hypothesis is wrong or right, and then based on the result, you do another hypothesis. I hate this kind of research. I like a research in which you do a screening and you find something that works. And then you go back and try to understand how it works. And so in both cases, we started from screenings, one in vivo for cardioprotective proteins that work. We don't even know how FAM3B, for example, or we don't have an idea, but they work, they do work. And the same for this microRNA, we came from a screening. So I'm very confident that we have something that works in mice. I think we all can agree. I show you data that are indisputable. Translation in pigs and then in humans can be difficult, but they work in mice. They are very conserved. They are the cardioprotective proteins are 95% conserved in humans, and they work in human cells. The microRNAs are human, so they have the human sequence and work in mice. So it's possible that it won't work, that it will be toxic, that whatever, but I think that I'm very confident that something can be taken out from this.
Okay, so do you have a question? Yeah, sure.
Thank you. Thank you for your outstanding talk. Just a very short question, maybe. I was wondering if you studied the role on inflammation in your therapies.
No, but that's super important. Yeah, super important. Inflammation probably is a collective term, no? So we should study the role of individual inflammatory cells, individual macrophage subsets. There is evidence that in other organisms that regenerate macrophages, for example, certain subsets of macrophages are essential. If you deplete macrophages, you don't have cardiomyocyte proliferation. So that's super essential, I agree.
I have one question. Sorry. By the way, we are breaking the record for the longest discussion we ever had here. But it's so fascinating. But I agree with you. We should look at what works. Is there anything that already works on cardiomyocyte proliferation of all the armamentarium? Because I personally think in heart failure we have to come a long way. You said that in absolute risk. In relative risk, it's more. I agree. But when we look at 5 or 6 drugs, we add 5 to 10 years of life expectancy in a 55 to 60-year-old heart failure patient. Now, that's not nothing. And compared to other fields in cardiology, that's actually a lot. So there's a lot that works. Is there anything that works into your concept that you are aware of? I'm not, but you would know.
No, there is nothing for cardiac regeneration. This is I think that our paper in Nature 2012 was the first demonstration that you can achieve cardiomyocyte regeneration by delivering something exogenous. There are plenty of there's plenty of indirect evidence that you can achieve adult regeneration in mouse models, transgenic mouse models, in which you activate a gene at a certain point in condition of established heart failure can regenerate completely. For example, there is a mouse model that I love, developed by Jim Martin in Houston, which he activates a gene in the hippo pathway transgenically. So basically, he gives Cree at a certain point, and he does this in mice in which there is already fibrosis. So basically, like some of the controls we had in which there is a big scar, and then he activates the gene in the residual cardiomyocytes, and he sees that these hearts regenerate and reabsorb the scar, which is not therapy, but tells that you can get there, provided that obviously the road is very long to this.
I never would have thought that to treat hypertrophic cardiomyopathy the way we've been treating it now.
Yeah.
In any time soon.
And
Nutritional drugs and amyloid are already there.
Yeah.
And they work. And not in all, but
Yeah.
they do work. So we've come a long way in the last 5 years. And this is the journey. I'm just coming back for Lisbon. You've seen the New England yesterday with in hypertrophy. This is happening. You know, I think we are more optimistic than we were probably 10 years ago when you started it. It was following your work all these years. You are a hope because if that one works, that would revolutionize heart failure in cardiology.
The excitement is contribute to this revolution, obviously. Not forgetting that it is not an easy task. It is not an easy task. But if there is a historical moment when this can be done, it is this one, because we have all the important tools. We have the genes, we have the tools to study them, we have the tools for delivery. We know better how to perform experiments in small, large animals, models, and so on. So if it doesn't work, if perhaps it will not work, perhaps not through this way, but I think that in this area, cardiac regeneration will happen, and it will not happen through patches or through the implantation of cells, but will happen by trying to mimic what stops happening at birth for some reasons, and just reinstalling that biological program.
All that I get, I know that I know nothing. The one thing is, I still don't get my questions answered that I've been asking myself over decades. Why is it that the heart is the only organ we know that doesn't really have tumors? You know, sarcoma.
Fantastic question. It's one of my favorites.
Yeah. You know, it doesn't. So why?
Yeah.
And why? And what does it tell us? And what is the switch? Congenital heart disease is for us insightful for a lot of things. What is that switch when you make your first breath that switches on the heart and then you stop regeneration? It was, in the end, the good things have to make sense. They don't come out of the blue. Yeah. It has to. There's a reason for this.
No, the reason, the evolutionary reason, I think it is quite understandable. I mean, evolution is driven by DNA, by our genes and not by our body. And our main purpose at birth is to cope with maintaining the individual alive. And to do this, you have to pump against pressure. And so you build up sarcomeres. You build a heart that certainly will take you up to 40, 50 years, which is more than enough time to have passed your genes to your progeny. And then if you have a myocardial infarction, you lose cardiomyocytes, you don't have regeneration, there is no evolutionary pressure for that. No? So I think that this is understandable, and this is the reason. Not only the cardiac tumors are very rare, but we were discussing with Francesco this morning, if you take tumor cells and you implant them, same tumor cells in the skeletal muscle in the limb, these cells, syngenic, grow enormously and eat up the muscle, destroy the muscle. You implant the same tumor cells in the heart, they don't grow. I was telling Francesco that we developed a few years ago AAV vectors with VEGF-465, the strongly angiogenic isoform. We injected it in the skeletal muscle, waited 1 month, and then look at the histology, there was a mess. So there were a lot of endothelial cell proliferation, big, almost hemangiomas, arterioles here and there, really a mess. I can show you the picture later. You implant the same, you inject the same vector in the heart, almost nothing. So the heart is an anti-proliferative environment. There is something that blocks proliferation in the heart, which could be, one doesn't have to think of a secretive factor, but it can be mechanosensing, no? Because the same mechanosensing. So this is the way I think it. At birth, you start having this afterload immediately after birth. And this is sensed by cardiomyocytes. And this sensing translates into stop proliferating, build up sarcomeres, and so increase in hypertrophy. But this sensing could be a sensing for all the cells in the heart, and including the cancer cells that you put there. Then, what is the sensing, the molecular sensor, and how this is translated? This is something that part of my laboratory is working on that, because you have the signal, then we can modulate the signal. But this is also probably why if you unload patients, they do better. This is why if you look at proliferation of cardiomyocytes in people with VAD, they do better. Some patients that are put on VAD before, as a bridge to cardiac transplantation, you remove the VAD and they don't need cardiac transplantation anymore. So I believe that is critical is understanding sensing of load.
I never thought about that, but it's fascinating. I love that. You know, this is,
we have some experiments planned with Francesco on this.
This is really,
stay tuned.
We have to close it, so we have to go to the restaurant.
Yeah, that was the longest.
It's so good because you tell us things that I sometimes, I feel myself running against the wall all the time. And this is actually a great concept of depression. I didn't really think about that. Not that we wouldn't be interested in depression, but I didn't make that leap yet. I learned something. Yeah.
There are so many questions, but I would not ask another question for sure. So on behalf of our cardiology, on behalf of CDEC, we thank you so much for being with us and for your outstanding lecture.
Thank you for inviting me. A pleasure.
What's the treat?
Oh, thank you.